4 weeks and then surgery. And
Okay.
rehab and graduate.
All right, guys. Camera's rolling. It's Wednesday, October 3rd. It's actually Raiders.
Is it on?
Yeah, it's on. Normally here's the date and the name of the class. It is AP Biology class. So anyway, hey, here's what we're going to do today during Raider time. We're not actually going to do a lab, but we're going to prepare to do a lab for next week. The reason I say that is one of the techniques is apparently kind of tricky. So I think we're just going to kind of use this time to fine-tune that one technique. So next week we can do this photosynthesis lab and be ready to roll. So that's what we'll do in a minute. But before I begin, I've been getting a couple questions from people, and it's actually really, really good about how to do your lab write-ups. Now, the lab write-ups will definitely, in your black and white notebooks, will follow the same format you guys are familiar with one exception. In your AP books, at the end of just about every lab are these analyzing results questions. Okay, so the lab that I know you guys are probably working on right now is the cell respiration lab. So if you turn to page 80 in your AP book, you will see 4 questions there under analyzing results. I would like you, yes, to answer those questions. I think those are going to be questions that the AP exam will want you to know. So those will act. I want you to answer those as well. Now, I think it makes sense to put them in your discussion sections of your lab write-ups. Hopefully, they won't be terribly too time-consuming. Don't worry about writing a book for each question. Just answer the question as clearly, concisely as you can. So there's 4 questions there. Now, be careful because that particular lab had a part 2. You see right below it, option 2. We didn't do that part. So obviously, there is no need to do the analyzing results questions that are on page 82 that go with that portion of the lab. So,
are you going to want us to restate the question?
Answer the question, or say the question and the answer.
Make them complete sentences, yes, because the AP exam wants complete sentences. Do the best you can, yeah, but don't be terribly worried. I mean, I'll have a general idea, but definitely complete sentences. Yeah, it's probably a good idea to restate it, but don't go too nuts with words. Any other questions about that? That's a good one. These are good ones. Again, remember, I'll grade those lab notebooks before the end of the first 9 weeks, so we can have that grade in the system. Hopefully, you know what? I don't even know if this other lab that's in there, the artificial selection lab, I highly doubt that one will even be completed by the end of the 9 weeks. So this might be the only lab that is in there, as well as this photosynthesis lab that we're kind of preparing for. So
Are we having a test before the...
Yes, you'll probably have a test sometime next week. So there will be a couple more grades before the end of the 9 weeks. Most likely 1 test, and then your lab notebook, which will consist of the cell respiration lab and the photosynthesis lab that we will do next week. That's kind of a tentative game plan. So, yeah, great. Jonathan?
Grady's not here.
I know. Grady's not here. This is why we're rolling for him. Okay.
The first question here, when it talks about use your data table to construct a graph.
Yep.
And the data table is the measure of the distance the fluid has moved at various intervals. I don't think we recorded that data.
Oh, that's right. We only took a beginning and an end, didn't we?
So do we still...
Oh, that's right.
We can just do 3 and 4, right?
Because we don't have any of that.
Yeah, we don't have that data for 1 and 2, do we? Because we didn't take it in intervals. Correct. I guess you guys can only do 3 and 4, can't you? It doesn't even make sense to do 1 and 2. We don't have that data. My bad. That makes it even easier, doesn't it? I'm trying to think of other questions. There were some other questions in that same lab in the beginning where it talks about, I tell you what, to make it up, I actually think it would not be a bad idea to, on page 73, I know you were probably supposed to do these questions in advance. Why don't we do this? I honestly don't want you guys to do this for busy work, but I think it's really worth your while to do questions 1 through 6 as well that talk about gas laws a little bit. Again, guys, I'm not looking for horribly long answers, but I'm looking for you guys to understand, those questions. A couple sentences, maybe we'll answer each of those questions. Put those as well. I know it's probably not ideal to put them in a discussion section, but let's put those there anyway. Let's put all those questions, 1 through 6 on page 73, and 3 and 4 on page 80, into your discussion section when you do this lab.
You just put like the number and
That's fine. I'll figure it out, yeah. Maybe do 1 through 6 and then 3 and 4, and I'll figure it out. Now let's do that. Like I said, I think that is for you. I think those are the questions that I know the AP exam will be turning to for guidance when they form their questions on the test. I would not be surprised, so it's worth your while doing. Good, yeah, next, very good. All right. Okay, any more questions on cell respiration lab? Again, that clears up a lot of stuff. That might be the only lab in this photosynthesis one that I will be grading here short term. All right, so then in your books, if you guys could turn to the photosynthesis lab, which begins on page 61. Here's what I actually just want to do. I'm going to, be a little boring and read here, but it won't take very long. We're kind of just prepping for this lab we'll do next Wednesday, but it's always a good idea to know what you're doing before you actually do a lab. And then we'll get to the fun part where we get to mutilate this ivy plant in front of me. So, all right, before we do that, photosynthesis. I'm going to read, follow along. Photosynthesis, I'm under background. Photosynthesis fuels ecosystems and replenishes the Earth's atmosphere with oxygen. Like all enzyme-driven reactions, the rate of photosynthesis can be measured by either the disappearance of substrate or the accumulation of product. Okay, so this is kind of similar to our cell respiration lab a little bit. They give you the equation there for photosynthesis, which you guys should be very familiar with at this point. And it asks you, what could you measure to determine the rate of photosynthesis? The production of O2, okay, or the consumption of CO2. Okay. So in this investigation, the last sentence, you will use a system that measures the accumulation of oxygen. Okay, remember in the last lab we did with cell respiration, we made those little microrespirometers, and we were measuring the differences in pressure. We were measuring basically how the consumption of oxygen. We were basically measuring with those little devices oxygen being used. So that's why we kind of saw this shrinking pressure. Anyway, this lab's going to be a little bit different, but let's continue. But we're still going to be measuring oxygen. Okay, but it's going to be very much more of an indirect measurement is what they're going to call it. The cellular respiration lab was a direct measurement. You actually got some data numbers. This lab's different. It's going to be an indirect lab. Yes, we're going to be measuring, it's going to ultimately measure kind of oxygen, but we're not going to get any number data for this one. It's very different. So next page, and then I'll be done. Because the spongy mesophyll layer of leaves shown above is normally infused with gases, O2 and CO2, leaves or discs cut from leaves normally float in water. Okay, and basically what they're saying is if I cut some of these leaves and I put them in a beaker of water, they're going to float because of that picture. Look at that picture, how many air spaces are caught in there. What would you predict about the density of the leaf disc if the gases are drawn from the spongy mesophyll layer by using a vacuum and replaced with water? How will that affect whether or not the leaf floats? If the leaf disc is placed in a solution with an alternate source of carbon dioxide in the form of bicarbonate ions, okay, then photosynthesis can occur in a sunken leaf disc. As photosynthesis proceeds, oxygen accumulates in the air spaces of the spongy mesophyll, and the leaf disc will once again become buoyant and rise in a column of water. Therefore, the rate of photosynthesis can be indirectly measured by the rate of rise in the leaf discs. Okay, stop right here. This is what this lab is saying. Here's what we're going to do. We are going to take little leaf parts, discs, that we're going to cut out using a hole punch. Okay. Now, just like the picture above, that little disc is going to have a lot of air spaces in it. Okay, so that's why if we put that in water, it's just going to float at the top. So what our first step needs to be in this lab, and that's the tricky part of the lab that I want to do today, is we've got to remove those air spaces from those little discs. And how are we going to do that? We're basically going to kind of create a vacuum. And I'll work on that technique in a minute. But once we remove the air spaces, okay, once we remove all the CO2, all the oxygen, all of that out of that area, now what was going to happen is we are going to take those leaf discs and we are going to put them in an environment where they are going to perform photosynthesis. And as you guys know, when you perform photosynthesis, one of the products you're going to make is oxygen. Well, oxygen is a gas. So what's going to happen is those leaf discs that are deflated because we just sucked out the air from them are going to gradually, as more photosynthesis goes on, fill up with oxygen once again, and eventually those discs will float to the surface. We are literally going to measure how long in time it takes for that process to occur. They mentioned these bicarbonate ions in here. Okay, those leaf discs, once we suck the air out of them, are going to be at the bottom of the beaker. Basically, we're going to put them in a solution with bicarbonate ions. That's going to be the source of carbon dioxide. Remember, photosynthesis is not going to take place unless there's water and carbon dioxide. Those are the two initial ingredients. Our source of carbon dioxide are going to be these bicarbonate ions. Literally, it's oh my gosh, it's tell me what this is.
Baking soda.
Thank you, baking soda. Oh yeah. Baking soda. Baking soda is filled. If you put that in water in a solution, it's basically going to that's going to be the source of carbon dioxide. You need carbon dioxide. You need water. Photosynthesis is going to occur. When photosynthesis occurs, those little discs are going to fill with oxygen. They'll eventually float to the surface. But there's not going to be any number measurements. Well, I guess with the exception of a time, how long it takes for that to happen. We'll have a couple of groups. Okay, one we're going to put under light, I believe, and one we're not going to put on. No, we're going to put both of them under light. But here's going to be the difference. One of them is going to be filled with oh my gosh, wait a minute. This is different. I believe we're going to have, no, I think they're both going to be placed under a light source. I'm trying to figure out the difference between the two control groups that they want you to do. I haven't read that far yet. Oh well, we're not going to get that far yet anyway. For some reason I thought it was light, but I'm beginning to think it's not a difference in light. They're changing some other variable in this particular lab. Anyway, no problem. I'm almost done, but that's the general gist. However, there's more. Oh, but I do want to read here. I'm still in that same paragraph at the word however. I'm about a little more than halfway through that paragraph on page 62. It says, however, there is more going on in the leaf than that. You must also remember that cellular respiration is taking place at the same time as photosynthesis in plant leaves. Remember that plant cells have mitochondria too. What else could be going on that might affect this process? Aerobic respiration will consume oxygen that has accumulated in the mesophyll. Consequently, the two processes counter each other with respect to the accumulation of oxygen in the air spaces of the spongy mesophyll. So now you have a more robust measurement tool. The buoyancy of the leaf disc is actually an indirect measurement of the net rate of photosynthesis. They're basically saying you have to call this lab an indirect measurement because you're not actually getting measurements as far as like we did in the cell respiration lab. Now, granted, in the cell respiration lab, remember we used KOH to get rid of one of the gases? We're not doing that here, which is going to be interesting, but that's one big difference between that lab and this lab is we understand that even though oxygen is being produced, some of that oxygen that we're producing is actually going to get consumed at the same time. But again, since this is an indirect measurement, it's not going to affect any data. Okay, here's what I want you guys to do. This is the fun part. I've been wanting to try this, and I wanted you guys to try it. This is what you guys need. I don't think I have enough syringes for everyone. I have enough. I brought 1, 2, 3, 4, 5. I'll tell you what, if you guys can share one syringe with 2 people, I think that'll work. So if we just kind of use 3 of them. These things are sort of kind of pricey, and since this is kind of a trial. What you need to do, take out a syringe, you need to take a hole punch, a single hole punch, and I have them on the table there. And you literally need to cut little circles of ivy leaves from my plant here. What I'm going to do though is cut some of these branches. Let's use the ones that I cut. Believe it or not, guys, I bought these plants over the summer, not to have a pretty classroom house plant, but for this lab, because I knew it needed ivy leaves. So I have another one in the back for if we need it, but this was actually used, this is supposed to be used for this experiment only. Cut out maybe 5, 6 of these little discs using the hole punch. Put them in there, put them in your little syringe here, okay? Then you're gonna put the plunger back in, but try not to crush them too bad. Then you're gonna come over here to the beaker over here. This beaker is filled with just a little bit of soapy water, very little soap. You'll suck up some of the liquid, probably to where it's mostly full. Okay, so I have pretty much a full syringe full of that water. Your leaf discs will be in there, but what you're gonna notice is those leaf discs are gonna pretty much be floating to the top. You don't want them to float. Remember, the whole step 1 of this lab is to basically get them to sink. So they say to do this. They say the best way to do this is to put your thumb over the end. Ooh, and these aren't the most ideal ones because it's going to be hard to do that. Put your thumb over the end, and you'll have to kind of hold it tight, and kind of squeeze, and basically you're pressurizing the inside of this thing. And if you do that and hold it for a few seconds, and then shake it up, do it again, shake it up, and do it again, theoretically, the disc should drop to the bottom. Basically, you're sucking out all the air, you're vacuuming out all the air by creating kind of a big pressure, by creating pressure on the inside. I don't know how well this is supposed to work. That's kind of why this is kind of a first go at it. So we'll see how it goes, and we'll see if we can get them to drop. Now, one word of caution: if you can't get them to sink within the first 3 or 4 tries, they basically say that it might just be a shot deal because you're going to basically damage the leaf discs if you keep doing this for like 10 times in a row, kind of deal. So they say try 3, 4 times, and if that doesn't work, totally get new leaf discs and try over again. So that's kind of what we're doing. Let's see if we can make, the whole point of the next 5 minutes, 10 minutes is to make those leaves drop. All right, let's kind of get to work and see if we guys can have any fluff on this. I'll just while we're playing.